Hello and welcome to another edition of Zog Science. Today we're going to be discussing meiosis and sexual reproduction. So, you've learned a little bit about genetics, right? So the question now is, how are genes passed from parent to offspring, okay? And there are two main ways. The first is through asexual reproduction. And asexual reproduction is a process where a single parent reproduces by itself. And when this happens, this is going to result in genetically identical offspring. This is mostly done by single-celled organisms. There are some colonial organisms that might do this, but mostly it's going to be bacteria and protists that are going to be going through asexual reproduction. Some plants can do asexual reproduction as needed, but the problem is that if you're not as genetically diverse, you have less of a chance of being able to respond to environmental conditions and evolving. The second way that we can go through reproduction is sexual reproduction. This is where two cells from different parents are going to unite to produce a new offspring. Okay, and this process is used to create genetically different gametes, which are those single cells from the parents that are then going to combine together to produce a genetically unique offspring. And this is why you do not look like your parents. You know, animals don't necessarily look like their parents as well. They're genetically different because we've got these unique gametes, these sex cells that are going to be combining together. So what is meiosis? Meiosis is the cell division that is going to produce those gametes. In this case, it's going to produce 4 genetically different haploid cells. Okay, what does it mean for a cell to be haploid? Well, in our normal body cells, we have 46 chromosomes, 23 pairs. That is considered a diploid cell. It has 2 sets of chromosomes, one that you got from your mother and one that you got from your father. A haploid cell only has 1 set of chromosomes. Okay, so it only would have, in the case of humans, 23 chromosomes. So meiosis is used to produce those haploid gametes, either sperm or eggs, that are then going to be used when you reproduce. Human gametes, when the process of meiosis occurs, we produce 4 equally sized sperm cells for men. However, in women, you actually only produce 1 egg cell and then 3 polar bodies, which are basically kind of small cells that eventually kind of disintegrate and go away and are not used in the process. So the phases of meiosis. Okay, meiosis is similar to mitosis, but it is not the same. Mitosis, remember, we've got each of the phases occurring once: prophase, metaphase, anaphase, telophase. But in meiosis, each phase occurs twice. So we actually get two kind of stages: meiosis I and meiosis II. Meiosis I is going to produce 2 diploid daughter cells. At this point, they are already genetically different from each other. And we'll kind of step through the process, each of these steps here in a moment. And then in mitosis II, those diploid daughter cells are going to divide again to make 4 haploid cells. So first of all, as always, we're going to start off with interphase, right? We've got the DNA. It's going to replicate, okay, right? At this point, it is chromatin. We also need to have the cell grow and go through all of the other phases in order to make sure that we have enough material for 4 different cells at the end. We then have prophase I. The DNA is going to condense to form chromosomes just like in mitosis. However, at this time, our homologous chromosome pairs are going to come together. So what happens is that both of your chromosome 1s replicate, right? And then they line up next to one another, and we have this process called crossing over, which occurs, okay? So the homologous chromosomes basically are exchange information. So homologous means same information. That's what logous means, information, and homo is same. So basically, you can have different combinations of genes that are going to be produced through this process of crossing over. So what that looks like a little bit more closely, your paternal and maternal chromosomes will cross over with one another, and so you can get pieces that will jump from one chromosome to the other. And this is part of the reason why you are able to have some traits from your mother and some traits from your father, because your chromosomes actually end up being a mix of the two. So metaphase I, the homologous pairs line up along the center of the cell. So remember, in mitosis metaphase, these chromosomes, the sister chromatids, would have all lined up along the middle of the of the cell, right next to one another, and then the centromeres would have broken apart. Here, the centromeres are actually going to stay intact, and we're just going to have the pairs move away from one another during anaphase 1. Okay, so the homologous pairs are going to be separated, moving to the opposite ends of the cell. Telophase 1, just like in mitosis, the nuclear membrane is going to reappear, spindle fibers disappear. We're going to reform those two nuclei. Then we've got cytokinesis. The two cells are going to separate. Okay. At this point, meiosis I is finished, and now we go into meiosis II. Okay. So prophase 2, the two haploid cells again are going to begin to divide. Prophase 2 really doesn't have that much going on with it. Just because there's no condensing that needs to occur, the chromosomes you can already see. Then we go through metaphase 2. So now this is going to be kind of more like the metaphase from mitosis. So the chromosomes, the sets of the sister chromatids, are going to line up along the cell. And then just like in mitosis, now in anaphase 2, the centromeres are going to split, right? And the sister chromatids are going to be pulled apart. Okay, and what this is going to do during telophase 2, they split. In cytokinesis, they completely finish that splitting. We end up with 4 genetically different haploid cells, and you can kind of see here that we've got ones that have certain pieces, okay, right, because there was crossing over that occurred. So each of these is different in terms of the genes that it has inherited. If you would like to take a look at a little bit more detailed animation of that, you can click on the link right here. So, comparing mitosis and meiosis, all right? They're similar, but there are definitely some differences, okay? Mitosis is only 1 cycle of cell division, whereas meiosis involves 2 cycles of cell division, because we need to divide twice in order to be able to get to 4 cells. Mitosis only occurs in body cells, which are also known as your somatic cells. That's basically everything except for your sex cells, or the gametes. There's no crossing over the chromosomes in mitosis, which is going to limit the genetic diversity, whereas crossing over does occur during meiosis during prophase 1. Mitosis results in 2 genetically identical diploid cells, whereas in meiosis results in 4 genetically different haploid cells. Finally, mitosis during metaphase, 1 duplicated chromosome per spindle fiber, right? So what that means is that your sister chromatids, you have the spindle fibers attaching to each sides, and they're going to break apart. Whereas in meiosis metaphase 1, 2 duplicated chromosomes per spindle fiber, so you actually have the homologous chromosomes lined up that are then going to be pulled apart from one another. No centromeres break during metaphase 1. All right. If you'd like to see a silly video, it's the volume is a little loud on this, so just be careful if you want to click on that link. All right. Thanks for watching, and I will see you all next time.